So first of all, I want to extend my thanks to Patricia and the rest of the Global Health team for the invitation to speak here today. I've presented in this forum before, and I previously spoke on the, again, as Patricia had mentioned, on iron deficiency and how it impacts fetal growth and developmental trajectories. And I thought today it would be an ideal opportunity to talk about some of our work on sepsis. Let's see here. Oh, there we go. So what we thought we would do is we would split this presentation. I'll speak first. I'll talk a little bit about the epidemiology of sepsis, and then talk in the second half about Sepsis Canada and the National Preclinical Sepsis Platform. And then rather than hogging all the attention, I thought then Faru could speak a little bit about about her work that focuses on the cardiovascular dysfunction that occurs as sepsis progresses to septic shock. Okay, so sepsis has undergone a couple of revisions over the course of the last 5 years or so, and the most recent definition, which was obtained at the third international consensus definitions for sepsis and septic shock, which is known as Sepsis 3. So sepsis is defined as a life-threatening organ dysfunction caused by dysregulated host response to infection. Okay, and so there's really essentially two aspects to this. The first is that by necessity, it's caused by an infection, but really what distinguishes sepsis from other straightforward infection is the presence of this organ dysfunction, okay? And so sepsis in its own right is a medical emergency, and it's associated with a high mortality, but if it progresses to septic shock, it's much, much worse. So septic shock is defined as a subtype of sepsis that's characterized by both profound cardiovascular, metabolic, and immune function. And again, it's associated with a much higher mortality, often because you end up with circulatory collapse, and oftentimes this cardiovascular decline is refractory to treatment. Okay. So this is the only slide I'll show on the pathophysiology of sepsis. On the left, I've got a more complex diagram, and then essentially a more simplified version that says the same thing on the right here. But essentially, sepsis is caused by an initial infection, a trigger, which ultimately leads to an exacerbated response. And so typically that looks like an exaggerated inflammatory response, but it's also characterized by immune suppression at the same time. Okay, and there's a widespread increase of reactive oxygen species throughout the body. And over time, this inflammation, oxidative stress can lead to a number of systemic deficiencies. So it's characterized by eventual coagulopathy, metabolic derangements, both on the systemic level as well as the subcellular level, and cardiovascular decline. And over time, if this is unimpeded, this will eventually progress to direct organ damage, as well as ischemia, which in turn can exacerbate the organ dysfunction, and this can ultimately culminate into death. Now, look at the left side, you'll see that in terms of the sensors and effector cells, almost every cell type that you can think of is implicated in the pathophysiology at some point. There's a number of mediators and biomarkers. A lot of these are cytokines and so forth. And importantly, ultimately, all of the organs of the body can be affected by this. And this really presents a lot of inherent challenges, because it means that not only is the pathophysiology, but also the clinical presentation tends to be very, very heterogeneous. Okay, and this produces a lot of challenges in dealing, diagnosing, and treating sepsis. Okay, but we do know that since the initial factor, or the cause, is infection, we certainly know that the sooner that we can get the infection under control, so timely intervention with antibiotics and source control, the better the prognosis and the better the management. Okay, and so because of that, there's been an uptick in awareness campaigns that are focused on, among the population, increasing the awareness of some of the major symptoms of sepsis. So, for example, very hot or very cold, confusion, disorientation, fast breathing, and this characteristic unwell feeling, or this extreme pain of worst feeling ever. Here on the right, you can see a similar sort of campaign by Sepsis Alliance using the acronym TIME, and it tells you essentially focus on the temperature, infection, mental decline, and again, this feeling of extreme illness. Okay. So these are some of the statistics for the epidemiology of sepsis, and it actually turns out that it's very difficult to get an idea of how many incident cases and what the case fatality ratio of sepsis is. I'll get to that in just one second, but in the United States, it's believed that 1 to 3 million patients every year are diagnosed. And as I mentioned before, it's got a very high mortality rate. So looking at the entire spectrum of sepsis, the mortality is somewhere between 15 to 30%, but once it progresses to septic shock, again, that mortality skyrockets to 50%. Okay. And then, of course, you can imagine then with this, the economic burden is also enormous. So it turns out actually the 14 to 18 billion is probably a little bit of an underestimate. I think current estimates are somewhere between 20 to 25 billion US in direct costs. Now, on the right, you can see the leading causes of death in the US, and each one of these ribbons here accounts for 10,000 deaths per year. And what you can see is that sepsis is believed to account for more deaths than all of these other major health illnesses. And in fact, it gets pretty close to all of these combined. Okay. Now, one of the reasons why it's actually hard to get a handle on the incidence and case fatalities is because in many cases, sepsis is not considered the cause of death. In many cases, it's considered an intermediary cause. Okay, and so then the death won't necessarily be ascribed to sepsis per se, but other health issues related to that. So I'll give you a case in point of what I mean by that. So this was a study that was published in The Lancet, and you can see that this was published actually right at the end of March in 2020. So this is right in sort of the upswing of when during the I guess before it was the COVID-19 pandemic. So this was, I think at this point, was still a global health emergency. And this particular study looked at the clinical course and risk factors for mortality of adult patients with COVID-19. And this was done right in ground zero. Okay, it was a small retrospective study, and they looked at total 191 patients in both non-survivors and survivors. And the key thing I want to draw your attention to is that in terms of outcomes, if you look at the non-survivors, so those that ultimately succumbed to COVID-19, 100% of them developed sepsis. Okay. So this is what the clinical presentation looked like. So these non-survivors developed fever, cough, and of course they tested positive for COVID-19. But in the eventual decline, sepsis was the first to manifest, which ultimately led to this acute respiratory distress syndrome that we know very well, and other acute organ injury. And so it's generally understood now that the most severe cases of COVID-19 are of viral-induced septicemia. Okay, so but here we've got a huge death toll, but we don't talk about it as deaths due to sepsis. We talk about it as deaths secondary or due to complications of COVID-19. So that's one of the reasons why it's really sort of hard to get an idea of what the incidence and mortality is. But last year, there was a seminal publication that was published in The Lancet that investigated the global, regional, and national sepsis incidence and mortality. And so this was an analysis of the global burden of disease study. Okay. And ultimately, what the authors concluded was from almost 50 million incident cases of sepsis worldwide, 11 million, there was 11 million sepsis-related deaths. But according to their estimates, the authors estimate that 19.7% of all global deaths are attributed to sepsis, which again, in my mind, is nothing short of mind-boggling. So 1 in 5 deaths that's occurring in the world today is attributed to sepsis in some capacity. Okay. Now, if you probe, there's actually quite a lot of information in this paper and a lot of interesting data, and I want to share a couple of these slides with you. So same publication, this is the epidemiology of sepsis, and this is incident cases in 2017. And the first thing you'll notice is that you look at countries like Canada, European countries are doing quite well. But actually, if you look down here, you'll see that this is, even the lightest panel is sepsis incidents per 100,000. So in Canada, we're still talking somewhere between 30 to 60,000 cases per year, okay? But obviously you can see that there's a huge geographical disparity here with a lot of low and middle income countries, a lot of countries in sub-Saharan Africa are really shouldering the brunt of this global health issue, okay? And certainly there's a huge disparity here, but I think that's even more pronounced when you look at the next slide. And this is the fatalities, so the case fatality of 2017. And again, so this is looking, again, geographical distribution, but this is total deaths related to sepsis in percentage. And so we're in the category of 0 to 8%, but again, looking at the same countries, some of these countries in sub-Saharan Africa are looking 50 to 65% of all the deaths within that country are attributed to sepsis. Okay, so there's a number of reasons why this might be the case, and I'm going to talk about a couple of challenges that are inherent to working with sepsis. And this is by no means an exhaustive list, but I think they're important, or they're challenges that we really have to be cognizant of, and you know, to be able to move forward. So one of the first challenges is that there's a very, very high cost of clinical care. So again, US figures, but the average cost per day for a patient with sepsis, and again, this is an average, including sort of the least sick to the most sick, is upwards of $18,000 per day. And so that, you know, when you consider that these patients are going to be hospitalized for many days, the average cost per stay is $53,000. And again, this is probably a bit of an underestimate. So when you consider the annual health spending per capita among countries, the average low and middle income country is spending about 1/20th the amount that the average high income country is spending on healthcare. And with that, that means that there's invariably going to be shortages in staff, perhaps lacking expertise to deal with this medical emergency, shortages of medications and medical resources, lack of infrastructure, and reduced ICU capacity. And even in post-septic discharge, a lot of these patients need a number of ancillary medical services to prevent rehospitalization and to reduce the high mortality in that first year post-sepsis. And you can imagine a lot of these regions probably don't have access to those medical services. Okay? So that's the first challenge. Now, again, it's worth mentioning here that, you know, this lack of investment, or this low investment in health spending, is not unique to sepsis, but by virtue of the fact that sepsis has such a high cost associated with it, I think it really accentuates some of these disparities. Okay? The second challenge relates more to this heterogeneity of disease. And I alluded to this at the beginning, but both in terms of its pathophysiology as well as its clinical presentation. And a lot of the work that's being done, both preclinical and clinical, is being done in high-income countries. So this is just a map. This is a snapshot of 2018 showing the number of current open clinical trials testing therapeutics for sepsis. And so you can see a lot of these are actually being done in countries that would be considered abundance or high abundance, or resource abundant regions. Okay, whereas again, those regions that are really bearing the brunt of this global health issue are not being conducted there. And so one of the questions is, why might we assume that evidence obtained in high income or high resource settings may not be transferable to those regions that have poor resources? And there's probably a number of reasons for this. So first of all, the populations may be very different. So there may be poor access to preventative health cares, a number of comorbidities, complications. There could be the presence of, for example, malnutrition, and there may be inherent differences just in the genetics, so that may influence immune responses. And that might mean that the population that we're looking at, the sick population, may be very different in some of these regions than in others. And of course, the pathogens, the vectors that are actually causing the infections, may be very different as well, not to mention the presence of antimicrobial resistance and so forth. So, so that's a big issue. The third issue doesn't really sort of provide any insights into the disparities between, say, high income and low and middle income countries, but I think it's an important issue that really has to be addressed. And this is really the enormous translation gap that happens from preclinical studies to bedside. So we're seeing a huge translational gap between bench to bedside. So This is a table basically showing a number of experimental agents that have been tested in clinical trials over the course of the last, say, 20 to 30 years. And you can see that the vast majority of these have been failures, and some of them have been absolutely monumental failures. Okay, successes really constitute the mainstay of treatment now, and there's a number of other therapeutics that are currently in the pipeline, but you know, there's not a lot of optimism with respect to these future therapeutics. And you know, again, it's worth mentioning here that this is not a problem that's unique to sepsis, but I think maybe perhaps more than other illnesses, it's an issue that we really have to confront if we're going to overcome some of the challenges associated with sepsis that I've presented here. Okay, so altogether, I've presented a very bleak view of this health issue. But I really do think that there is cause for hope. And, you know, I think necessity is the mother of invention. And in this particular case, it might be the mother of reinvention, because I think a lot of the challenges that I've talked about here have been a powerful impetus for the research community and the medical community to, you know, to approach sepsis, to approach research in a more communal and a more unified front. Okay, and so over the course of the last 5 to 10 years, there's been a resurgence of international collaborative efforts that are focused on understanding the pathophysiology of sepsis. And Canada's response to this is Sepsis Canada. So Sepsis Canada was founded in 2020, so just last year, and it was based largely off a large team grant, a $5 million team grant from the Canadian Institutes of Health Research. Okay, and Sepsis Canada is a multidisciplinary network really committed to understanding the causes of sepsis and improving the prevention, detection, management, and even rehabilitation through research. And it focuses on a number of areas. Again, and I'll talk a little bit about some of these. So essentially, Sepsis Canada is broken down into 3 teams, and there's a number of themes that each of these teams are addressing. And again, they span the gamut of sepsis-related research, so understanding the burden, the epidemiology, populations at risk. You know, there are some teams that are focused on creating a culture of awareness and those using evidence to inform practices and to improve clinical care. For my part, we're biomedical scientists, and so really mostly involved in Team 3. And so Team 3 is mostly involved in understanding the pathophysiology, the pathogenic mechanisms leading to sepsis, and then focused on developing novel intervention strategies. But although we've been doing this for some time, the real difference is that these teams now are approaching these problems in a more collaborative, in a unified method, okay? And that's really what I want to talk about right now. So one of the elements of Team 3 is the National Preclinical Sepsis Platform. And so this is a picture of us that was taken at Lake Louise. This was before social distancing was a thing. But this is a group of about 20 to 30 investigators. It's supported by Sepsis Canada, the Canadian Critical Care Translational Biology Group, and the Canadian Institutes of Health Research. And most of us are biomedical scientists, and really the NPSP is trying to tackle the 2 major issues. Again, most of our work is focused on understanding the complex and variable presentation, treatment response, and outcomes, and as well this complex pathophysiology. But again, one of the things, as I mentioned, that absolutely has to be addressed is this crisis of translation for preclinical sepsis research. And I think that confronting this, addressing this sort of elephant in the room is going to be critical to moving forward and actually making progress, okay? And so, you know, it begs a couple of obvious questions. Why do we have experimental derived therapies routinely used in clinical practice? You know, why are there such poor linkages between basic scientists, clinical researchers, and patients? And so these are some of the questions that we're really trying to address. And I guess in the last 5 minutes, I'll tell you about one initiative that I think is really interesting that gets to hopefully we'll start to address this issue. And of course, in the next part, Faru will talk a little bit about some of our work that focuses on the pathophysiology of sepsis. Okay, so one of the initiatives that the National Preclinical Sepsis Platform is doing is beginning to develop the platform for a preclinical multi-center study. And so this is exactly as you might expect. It's the, I guess, preclinical equivalent of the multi-center clinical trials, which is the gold standard in clinical research. And this is where we have multiple independent laboratories performing similar experiments using shared protocol. So typically the way we've approached our research is we develop our own animal model, we develop our own treatment protocols. And the challenges to this is that if multiple individual labs are doing it, it's very hard to consolidate this information and synthesize it, okay? Whereas the multi-center preclinical trials, there's involvement of multiple groups at different institutions, and we adopt a common and shared protocol, and everyone implements this protocol in tandem. And so by the end of this, hopefully, all of the data that we can collect will be interchangeable and will be much easier to synthesize in sort of a whole, in a holistic conceptual framework, okay? So obviously there's an increased cost with this. So if we've got, 6 individual laboratories performing the same experiment, huge increase in cost, what are the benefits to this? Well, of course, it increases generalizability and it tests reproducibility. This is inherent to the design because, again, all of these outcomes are going to be tested independently in all of these various centers. And because of that, it produces a more robust outcomes and database than single center studies. But perhaps most importantly, it arguably increases the efficiency and efficacy of preclinical translation. And to illustrate that, this is a nice systematic review that was published just last year. And essentially what this study did was they looked at the number of multi-center preclinical trials, okay? And there was 12 of them that they found. And these were preclinical studies that were trying to validate the positive outcomes that had been reported in single center studies. And so out of the 12, 4 of these multi-center preclinical trials were able to recapitulate what had been shown in their single center cohorts. But 2 of them had mixed results and a full half of them could not replicate what had been shown in single center studies. So this is important because if these single center studies had made the recommendation to take their therapeutic and move it on to clinical trials, without first of all being able to validate in a number of independent laboratories, this could be a huge waste of resources and time. And so instead of investing our time in these guys, it would be obviously much more efficient to invest our time in the ones that have the potential to actually be successful in clinical trials. Okay, so ultimately what we're trying to do is do a number of multi-clinical center or a multi-center preclinical trials. And the first step to this is actually developing a universal standardized model of sepsis. Okay. And so I think in the last couple of minutes here, I'll just talk about the model of sepsis that we're using, and this will actually segue really nicely into Farou's work. Okay. So for our model of sepsis, we use what's called the FIP model, or the fecal slurry-induced peritonitis model. And so this is an active model of infection. Okay. And essentially what we do is we take a number of donor animals and we collect the cecum, and then we harvest the contents within them. And then ultimately we homogenize them and we prepare this suspension. It's in a glucose solution. Okay. Then you make it homogeneous, and then you can aliquot it. And the nice thing is you freeze it, and then whenever you want to do a study, you take a single aliquot, and then you can take this fecal slurry, you inject it intraperitoneal, and then you induce sepsis. Okay. And so even though respiratory sources are the most common cause of sepsis, so typically secondary to infections like pneumonia, intra-abdominal sepsis, which is essentially what we're modeling, is the second most common cause of sepsis and is also incidentally the most deadly. Okay. Now, the nice thing about this model is that you can induce sepsis to whatever severity you would like. So you can add a small dose and cause a fairly modest degree of sepsis, or as Faru is going to show you, we add a fairly big dose and we can cause cardiovascular collapse within 6 hours. Okay. And so although that's a benefit to the model, it's also another challenge because this in itself introduces a great deal of heterogeneity as well. So how do you reconcile, how do you consolidate data from our lab group on the cardiovascular work in such a severe model? How do you compare that to, for example, the mitochondrial dysfunction that another group is studying when they're looking at a much less severe model? So it's really difficult to bring all of that data together, okay? So in this model, one of the nice things about it is I don't want to say it's the gold standard of models, but it certainly does recapitulate all the salient features of sepsis. So here we have the clinical score, which is basically a number of parameters based on behavior. And you can see that after you inject the fecal slurry, these animals get progressively sicker over the course of, say, 20 hours or so. And ultimately, over the course of 24 hours using this particular dose, we saw about a 50 to 60, or probably about a 60 to 70% mortality rate, okay? But again, higher doses, you can get much faster decline and vice versa. And of course, you get all the good stuff that you would expect with a good model of sepsis. So you get macrophage infiltration in the peritoneal cavity, markers of inflammation, and organ damage, okay? Now, this is the model that we've been using for a number of years, and one of the first things we did as the National Preclinical Sepsis Platform was to just compare the fecal slurries that were coming out of 2 laboratories. Again, in the interest of standardizing a model, we wanted things to be reproducible. And so what we did was we had our group, the University of Alberta, prepare a fecal slurry, and we did it in tandem with another group, which McMaster University. And so to do this, what we did was we ordered our animals from Charles River from the same litter. We had them shipped and delivered on the same day. The animals were not housed in our animal care facility for fear of, adopting a unique microbiome that's unique to the institution. We ordered all the same chemicals, and we even tried to prepare the fecal slurry over Skype so that if there were any inconsistencies or there other sort of things that were not clear, we could resolve them in real time. Ultimately, we weren't able to do them over Skype because of a number of complications, but ultimately we did our best we could to prepare these fecal slurries to make them identical. And so then after we had prepared these fecal slurries, we sent them off to the University of Calgary for a microbiome analysis. And here you can see the results from that. So this is the microbial taxonomy. And so these are our samples. We shipped 5 separate samples, and same with McMaster. I guess the first thing to notice is that the samples do look fairly consistent, but there is a number of differences. And in fact, when we did the principal components analysis, you can see that there's a very clear clustering from the samples that were prepared at our institution compared to McMaster University. So again, even in an attempt to standardize our protocols and make everything as repeatable as possible, we still end up with marked differences in the makeup of these fecal slurries, okay? So based on this data, we ultimately resolved among the PSP that only a single institution is going to prepare all the fecal slurries, and then once these are prepared, we're going to distribute them out to the other centers. And I'm not sure if we're the lucky ones or the unlucky ones, but we were the ones who were selected for preparing this fecal slurry. And ultimately, we'll be shipping them out to all these other centers across Canada. As I mentioned before, the first strategy is to characterize this model using predefined set points, inflammatory markers, markers of organ dysfunction, circulatory factors. And then once that's been characterized and replicated among all these different institutions, their individual expertise will sort of take over. And then we can start to explore, for example, the blood flow experiments that we want to do using the standardized model. So we think that this is going to be a new way of using a standardized model, but being able to synthesize and consolidate all the different data sets that are coming out using this one model. Okay, so I've talked long enough. So I'll just give you a brief interim summary and say that sepsis, again, is a complex disease entity with heterogeneous pathophysiology and clinical presentation. The challenges of managing and treating sepsis highlight really the disparities between resource-abundant and resource-poor settings. And what I think is an exciting initiative, Sepsis Canada is a collaborative and multidisciplinary research initiative aimed at improving the recognition, management, treatment, and recovery from sepsis. And so with that, I will stop sharing my screen and I will let Farouk take over.
Hello, everyone. My name is Farouk, and I'm a postdoctoral fellow working on the sepsis project in Dr. Burke's lab. In this part of the presentation, I'm going to talk about the importance of organ blood flow assessment in sepsis. As mentioned earlier, sepsis starts with infection, which causes a widespread oxidative stress and systemic inflammation in the body, which affects several organs and systems in the body and over time leading to reduction in the blood pressure, and eventually reduction in the organ blood flow, development of multi-organ damage, and death in septic shock patients. In clinic, several vasopressors are used to increase the blood pressure. But we believe that focusing only on the systemic hemodynamics to increase the blood pressure without considering the potential effect of such treatments on the organ blood flow can be misleading. The reason is that these drugs increase the blood pressure through increasing systemic vascular resistance and narrower blood vessels. Although it increases the blood pressure, it limits the blood flow through the vasculature into the organs. As an example, how these vasopressors can actually affect organ blood flow, we did an experiment. When we infused norepinephrine in the mice, you can see that it caused an increase in the blood pressure in the animals. However, it dramatically also affects the organ blood flow, especially in the kidney and gut. This clearly shows the importance of considering organ blood flow when any treatment or intervention for sepsis is used. In our laboratory, we have the infrastructure to study the changes in both systemic and regional hemodynamics in a murine model of sepsis. Therefore, we wanted to assess the changes in hemodynamics and see how the changes in blood pressure relate to changes in regional blood flow. We wanted to explore if vascular dysfunction occurs in all the vascular beds at the same time, and if not, which vascular beds are more susceptible. And finally, we wanted to know if triggering damaged vascular beds in sepsis can have survival benefits in this setting. So, as I mentioned, we use a murine model of sepsis. So, mice were anesthetized first, and hemodynamic assessments were done by inserting fiber optic pressure sensors in the right carotid artery of the animals for measurement of blood pressure and heart rate. Perivascular flow probes were put around the carotid artery, left carotid artery, superior mesenteric artery, and renal artery for measurement of blood flow to the brain, gut, and kidney. All these procedures were done by Sarah in our laboratory. And also, fluids and drugs were administered through the femoral vein. After instrumentation and stabilization of these animals, fecal slurry was injected intraperitoneally for induction of the sepsis in our model, while the control group received just a vehicle. And we looked at the changes in the hemodynamics over time before the cardiovascular collapse happened. So these graphs show changes in the systemic hemodynamics over time, which is shown in the x-axis, in both groups, in the control group, which is shown in the black color, and the septic mice, which is shown in the with the red color. The graph on the left shows mean arterial pressure, and as you can see that sepsis caused a gradual decrease in the blood pressure in these animals, where changes were noticeable at 2 hours after sepsis induction. The graph on the right shows changes in the heart rate in the animals, and as you can see, sepsis had an overall effect on increasing the heart rate in these animals. This is probably due to the compensation for the reduced blood pressure in these animals. We also looked at the changes in the regional blood flow over time, and the graph on the left shows the blood flow profile in the carotid artery, while the middle one shows the superior mesenteric artery blood flow profile, and the right one shows changes in the renal artery blood flow in the control and septic animals. While we observe reduction in the blood flow, in regional blood flow in all the studied vascular beds, we noticed a distinguished biphasic reduction in the blood flow in the carotid and renal artery, which shows a kind of autoregulatory mechanisms that tries to keep the blood flow to the most vital organs in the body, like brain and kidney. However, you see that with the progression of sepsis, these mechanisms are compromised and changes, reduction in the blood flow is observed around 2.5 and 3 hours after sepsis. In the SMA blood flow, however, we observe reduction in the blood flow almost immediately after induction of the sepsis, and it's a progressive reduction. So this shows that different mechanisms are involved and how the vascular beds are responding to the progression of sepsis to septic shock in terms of blood flow. While these graphs all show like the average blood flow profile in the vascular beds, we also wanted to look at the some mechanism behind the development of vascular dysfunction in sepsis. So to do that, we assess in vivo vascular reactivity in the live animals. So we used vasoactive bolus doses of vasoactive drugs at the baseline before induction of the sepsis, as well as every 30 minutes after sepsis induction to see how the responses are affected in both groups. So we use phenylephrine, which is a vasoconstrictor at 5 microgram per kg. At this dose, it causes a sharp transient increase in the blood pressure without affecting the heart rate. This increase in the blood pressure is resolved within 2 to 3 minutes, and the blood pressure returns to its normal level before drug administration. So this shows that any changes that occurs to the blood pressure, because it doesn't affect the heart rate, could be due to the effect on the vasculature and vascular function. Another drug that we used was sodium nitroprusside, which is a nitric oxide donor, and it causes vasodilation. At the 6 microgram per kg that we used, and at this dose, it also causes a transient reduction in the blood pressure, which resolves within 2 to 3 minutes. And it doesn't affect the heart rate. And again, it's another drug to kind of study the mechanism of vascular function or dysfunction as sepsis progresses to septic shock. So this graph shows the response, blood pressure or systemic hemodynamic response to phenylephrine in both groups over time. You can see that while at 0 time point, which is the baseline, the response is comparable in both control and septic animals, and it causes around 60 millimeter mercury increase in the blood pressure. Control animals more or less maintain this response over time. However, you can see a gradual decrease in this response in septic animals, which can point to the development of vascular dysfunction in these animals. So this is the systemic hemodynamic response to phenylephrine. We also were interested to see how blood flow changes, how blood flow responses change with the administration of phenylephrine over time. And we looked at the regional blood flow changes. And in that case, we observed very interesting and different story here. The changes in carotid blood flow in response to phenylephrine shown in the left graph. The middle one is for the superior mesenteric artery blood flow, and the one on the right is for the renal blood flow. And one thing that you can see here is that the effect of the the response of the vascular bed to the phenylephrine administration. While in the carotid artery, we observed a modest increase in the blood flow. You can see that in superior mesenteric artery as well as renal artery, there is a reduction in the blood flow, which is their physiological response. Interestingly, sepsis did not affect the blood flow response in the carotid artery in response to phenylephrine, and we didn't see any effect, and all the responses were comparable between the septic and control group. However, when we look at the superior mesenteric artery blood flow changes in response to phenylephrine, an immediate effect on the was observed. So 30 minutes after induction of the sepsis, the first time point we saw an increase in the blood flow, which is contrary to what we see in the vehicle group, like in the control group and their physiological response. And in the renal blood flow, we also observed changes around 3 hours after sepsis induction. So these graphs clearly show the heterogeneity of development of vascular dysfunction in sepsis and how different vascular beds are responding or affected by sepsis. We also, as I mentioned before, we also look at the effect of sodium nitroprusside, which is a nitric oxide donor, and it causes vasodilation, as you can see here, around 17 millimeter mercury reduction in the blood flow, blood pressure, was observed. And again, it was comparable at the baseline between two groups. The control group more or less maintained this response, and we observed a gradual decrease again in the blood pressure response to sodium nitroprusside in septic animals. We also looked again at the regional blood flow responses, and the same story was observed here. SNP caused a modest reduction in the blood flow in the carotid artery, while it increased the blood flow in the mesenteric artery and almost had no effect on the renal, or a little bit increase in the renal blood flow. Again, carotid blood flow changes were not affected by sepsis in both groups. And again, superior mesenteric artery was the first vascular bed to be affected by sepsis, where changes were observed 30 minutes after induction of the sepsis. Interestingly, for SNP, we also observed that renal blood flow changes were affected 1 hour after induction of the sepsis in SNP case. So this has another, this shows another kind of effect and shows that different mechanisms are involved in the progression of sepsis to septic shock in terms of vascular dysfunction. So we showed that vascular dysfunction occurs in sepsis before changes in systemic blood pressure and hemodynamics are evident. Organ blood flow patterns better reflect the cardiovascular decline in sepsis. And so this can point to the fact that maybe monitoring organ blood flow is useful in predicting the organ injuries that occur in sepsis or septic shock, such as acute kidney injury or necrosis in the gut. So as we show all these, we wanted to also explore if targeting specifically dysfunctional vascular bed can have survival benefit in septic mice. So we focus on nitric oxide, which is a potent vasodilator. Nitric oxide has lots of physiological effects, especially in increasing the vasodilation and organ blood flow. However, nitric oxide is also has a key role in the pathophysiology of sepsis. Under physiological conditions, nitric oxide activates the soluble guanylate cyclase in the vascular smooth muscle cells to produce cGMP, which has vasodilation, vasodilatory effect and increases the organ blood flow. However, in pathophysiological conditions associated with reactive oxygen species generation, when there is oxidative stress, like in sepsis, SGC is damaged. And this damaged form of SGC can no longer be activated by nitric oxide. This leads to this leads to the perturb of the downstream signaling of the SGC, and there's no cGMP, not enough cGMP production to induce the vasodilation, and therefore there is reduced organ blood flow. Sinaceguat is an SGC activator, which can selectively target this damaged form of SGC. So when targeting the damaged form of SGC and activating it, the downstream signaling can again be resumed, and cGMP is produced, and regional vasodilation, and causes increase in the organ blood flow. So to test the hypothesis that if intravenous administration of sinaceguat in septic mice can improve survival, we design an experiment where septic mice receive sinaceguat 30 minutes after induction of the sepsis, and we looked at the survival. We used 15 microgram per kg body weight of sinaceguat as preliminary experiment show that this dosage has the best survival benefit. Increasing the dosage of the sinaceguat dilutes the survival effect, and that is potentially due to the non-selective effect of sinaceguat at higher doses. When we use this dose in septic animals, we observed an increase in the survival rate of sepsis animals. So we show that sinaceguat administration improves survival. We also looked at the effect of sinaceguat administration on the SGC activity at the tissue level. For example, we showed that in the kidney, while sepsis caused SGC damage, as you can see in the sepsis, the level of damaged SGCs were increased. Sinaceguat administration reduced this damage form, and the level of damaged SGC were comparable between control, which are non-septic animals, and septic animals receiving sinaceguat. So this clearly shows that sinaceguat specifically targets the dysfunctional vasculature in sepsis. Currently, we are assessing if sinaceguat administration actually improves the blood flow, regional blood flow in the septic animals, to see if the observed survival effect is due to the improved regional blood flow in these animals. So we showed that blood flow assessments are critical in understanding the pathophysiology of sepsis. The circulatory dysfunction that occurs in sepsis is multifactorial, and probably different treatment strategies are required. We show that the SGC activator sinaceguat can have potential therapeutic benefits when it is used early after sepsis. So with that, I would like to thank all our collaborators, Dr. Kimberly Makala, Dr. Rachel Khadarouk, Dr. Frante Garagasin, our amazing technician, Ms. Sarah Panahi, and members of Bork Lab, as well as all the funding agencies who support our research. With that, I would like also to thank you all for listening to our presentation, and we're happy to take any questions you might have.